Hello everyone. So the next topic is about the Dynein. So Dynein, it is the one of the three cytoskeletal motor protein families which was first identified a half century ago, and it's named after the dyne, which is a unit of the force. So this is what is the dynein. So most of the motor proteins, they are fueled by ATP. We know that, and they are essential. What they're essential for? Essential for the movement, division, and special organization of the eukaryotic cells, as well as the generation of forces great enough to enable the transport of cellular cargo along what may be called the cytoskeleton highways. So the importance of these cytoskeleton motor proteins is seen where any dysfunction within them can result in a serious disease implication. So dynein, it carries out its duties as a protein complex. It is constructed around subunits, which is called as the heavy chains, and they are capable of generating the forces. So these chains, they are so named due to their hefty molecular masses and the motor domains, which is belonging to AAA plus superfamily, attached to the divergent amino as terminal tail domains. So these tail domains, they possess distinct properties with regards to oligomerization, and they allow for the binding of a variety of associated subunit types. So these subunits in turn are mediators for cargo interaction, binding, which may occur directly or may be achieved via adaptive routines. Now, what is the history of this dynein protein? So the protein which is responsible for the movement of cilia and flagella was being discovered, and it was named dynein in 1963. So 20 years later, there was a cytoplasmic dynein which had been suspected to exist since the discovery of a flagellar dynein and was isolated and identified. Now, talking about the structure of the dynein, then each molecule of the dynein motor is a complex protein assembly composed of many smaller polypeptide subunits. So, the cytoplasmic and ozonal dynein contains some of the same components, but they also contain some unique subunits as well. So if I talk about the cytoplasmic dynein, which has a molecular mass of about 1.5 megadaltons, it is a dimer of dimers containing approximately 12 polypeptide subunits, which is two identical heavy chains, that is 520 kilodaltons in mass, and it contains the ATPase activity, and thus they are responsible for generating movement along the microtubules, that is the 274 kilodalton intermediate chains, which are believed to anchor the dynein to its cargo, and 253 to 59 kilodaltons light intermediate chains, and several light chains. So this is the overall dynein cytoplasmic dynein introduction. Next is the functions of the dynein. So what are the functions of the dynein? They include, first of all, the dynein will perform a number of cytoplasmic cellular functions. It serves as the power behind the transport of a membrane-bound tubules and vesicles in conjugation with their occupant molecules. So this cargo is transported towards the minus ends of the microtubules. Organelles transported along the cytoskeleton highways include the lysosomes and endosomes, phagosomes, perisomes, melanosomes, mitochondria, lipid droplets, and endoplasmic reticulum vesicles that are headed for the Golgi. So this transportation can also be employed for the transmission of certain signals across cellular regions. Furthermore, some recent data suggests that dynein may be involved in the translocation of the proteins spanning the membrane and lipid bilayer signaling complexes. So dynein is also involved in the clearance of cellular debris that need to be degraded or recycled. For example, autophagosomes, they engulf proteins and organelles at distal neuronal uh, points, and they transport these cargo in a dynein dependent fashion towards the cell body for the processing. So in addition to these functions, dynein may play a role with certain viruses, albeit involuntary, in the migration to the nucleus of the cell. So there is an example of this, which could be the resourcefulness of the HIV, that is the humoimmunodeficiency virus, hijacking dynein to reach its intended nuclear designation. So, other additional functions of dynein include its capacity to exert tensions on structures within the cell. So, a force tugging on the microtubule networks can be generated and maintained by dynein proteins that are tethered within the cellular cortex. So dynein accomplishes this by one or of two ways, either by its migration towards the microtubules minus ends, or by coupling itself to a plus end of the microtubule, that is the disassembly. It can thus pull the cytoskeleton of microtubules towards a T cell immunological synapse, or it can pull the cytoskeleton of microtubules in migrating fibroblasts and neurons towards the leading edge. So these were all about the functions of the dynamic. So these are some of the references.